Alrighty, welcome back. We're going to pick up here talking about lymph nodes, a little bit about the structure and exactly how they're involved with the filtration of our lymphatic fluid. So, lymph nodes are small, little oval structures that are little, you can almost think of them as kind of beads on a chain. The chain being the long lymphatic vessel, and then these beads, the lymph nodes, kind of interspersed throughout. Now, you have a kind of an aggregation of these lymph nodes where multiple vessels are kind of coming to a single point. So like we said earlier, they are common in the cervical region, in the axillary region, as well as in the inguinal regions and also the pelvis. Now, what these nodes do is they're essentially a site for filtration of the lymph within the fluid, the lymph within the vessel. Whenever there is an active infection, our lymph nodes will generally swell because there is increased fluid. There are increased lymphocytes in that area, in that node. And again, we have our B cells in that node that whenever it encounters an antigen and ultimately has to become activated, it will then differentiate into a plasma cell, which we said secretes lots and lots, thousands and thousands of antibodies per second. So they're antibody factories, essentially. Now, here you can see our aggregations of nodes, so our inguinal nodes, our axillary nodes, and our cervical lymph nodes. These are very common areas, but again, remember that there are nodes really all throughout the body. So, the structure of a node, and we'll look at this in lab too, you'll see this in a lab setting. There's a capsule, which is just going to be the outermost layer, so you have a capsule of most organs. When we see the kidney, there's a fibrous capsule of the kidney. There are little projections of this capsule kind of into the lymph node that kind of divide it into sections or compartments, and these are going to be called trabeculae. So trabeculae is also a term that's thrown around when you talk about the skeletal system. So trabecular bone or spongy bone. It's this kind of lattice network like a bone. So trabeculae in this sense is just going to be this small little extension that's going to divide the node into different compartments. We have a cortex, which is an outer region consisting of T and B cells. We then have a more centrally located medulla, which largely has macrophages. And then we have these little sinuses that are little passageways from the cortex to the medulla that allow lymph to flow as it enters the lymph node. So a picture will make a little bit more sense. So what you have your capsule, which is this outermost region. You have the trabeculae, which are just extensions of the capsule in. You see a cortex, which is this outermost portion here, and then you have this more centrally located medulla. We'll talk about these vessels that are coming in and then those that are coming out, essentially, and leaving the lymph node as well. So you have these vessels that are coming into the lymph node, and these are going to be called afferent, or sometimes you'll hear them referred to as afferent lymphatic vessels. So they're afferent, they're arriving. So think of the A in afferent as standing for arriving at the lymph node. Okay, it will then enter the lymph will then enter the subcapsular space. So the subcapsular space is really just the area just immediately underneath the capsule, and then they'll enter these sinuses that will ultimately flow and lead into the medulla. And then when all of the lymph flows through the sinuses to the medulla, it's filtered, and then it will exit. So think of the E in this next term called efferent or efferent. Think of the E standing for exit. It will exit the lymph node, and it will exit at a specific location called the hilum. So the hilum is not a term that's specific to the lymph node. You have a hilum of the kidney, you have a hilum of your lungs. The hilum just means it's this area where things are entering and exiting a structure. So for the lymph node, when we look at a picture, this region here is going to be the hilum, where you have vessels coming in and coming out. So you have arteries and veins also coming here as well. Now, I want you to note the difference in the number of afferent vessels versus efferent vessels. Are there more afferent lymphatic vessels, or are there more efferent lymphatic vessels? So hopefully looking at this picture, and most drawings will depict this, there are more afferent vessels than there are efferent vessels. So think about what this would do to the flow of lymph as it enters the lymph node and then as it's leaving. So what would happen to the flow if there are more afferent than there are efferent? Hopefully you're thinking that if you're bringing lots of fluid in, lymphatic fluid into the lymph node, and you only have two ways to get it out, it's really going to slow it down. So think about a large, so there are four vessels here. Think about a large 4-lane highway. And then all of a sudden, those 4 lanes merge into 2. What's going to happen to the traffic? Well, the traffic's going to slow down drastically. Same idea here with our lymph node, with our vessels. You have 1, 2, 3, 4 afferent vessels and only 2 efferent vessels. So the fluid will come in, flow from the subcapsular sinus through the cortex and into the medulla, and it will slow down as it's leaving the efferent vessels. So this allows the fluid to have more time within the lymph node so that there's more opportunity for the filtration and to target and neutralize any foreign pathogen or foreign material that happens to be in the fluid. So there are more afferent vessels than there are efferent vessels. So the last section we'll talk about here are lymphatic organs. And so some of these we've talked about. So the spleen is the first one we'll hit on. It is a very large lymphatic organ, lymphoid organ. It's going to be located in our left upper quadrant. And it actually contains a decent amount of blood at any time. So we call it a blood reservoir. So let me switch to a laser so it's a little bit easier for you to see. It's going to be a blood reservoir. So what a blood reservoir means when we say this, it's a site, the spleen is, where lots of blood can be contained at any time. So you can imagine if you were to have an injury here and you were to rupture your spleen, or oftentimes it's common in stabbings, if someone is stabbed in their left upper quadrant, you can pierce your spleen and you can actually bleed a very significant amount because there's lots of blood there. It's also a site where we filter different old blood, and we filter out red blood cells, so our erythrocytes. And in the fetus and fetal development, it's actually involved with the production of red blood cells and white blood cells. But the primary thing I want you to focus on is that it's going to filter our blood with macrophages. We're going to destroy and recycle old red blood cells, and it's a common location for lots of blood to be really at any given time. So within the spleen, when you look at it histologically, you can see different components. There's going to be a white pulp, a part that we'll call white pulp, and then a part that we'll call red pulp. Now the names white and red we get for specific reasons. White pulp contains lots of lymphocytes, and lymphocytes are going to be a type of leukocyte, and leukocytes are white blood cells. So we call this the white pulp. And then there's a red pulp, and it's red because it contains lots of blood and red blood cells. So this is where filtration is actually occurring. This is where the blood is being directed to as it's being filtered. So here's a gross anatomical view. So this would be, if you see it on the left here, you know you're looking at a posterior view. And then you can see this large organ over here to the left. It's actually in an actual human, it's actually very unimpressive. There's not a whole lot to look at. It's just kind of this large mass of tissue, but it's highly vascularized. So you can see lots of blood vessels here. So when we get to blood vessels, when you look at them in a lab setting, this large vessel back here behind the stomach, this is going to be your splenic artery, and it will drift over here to the left and provide blood to that spleen. Now, here you can see a histological view. Again, this is on Histology Guide. A histological view of our spleen. So what you see here in the middle, you see our white pulp. It's a little lighter in color. So this is where we have a lot of our T and B cells. And then you can see your red pulp around the side. This is where most of the blood and the red blood cells will be. And so it is aptly named red pulp. And then again, we have white pulp that contains our lymphocytes, so our leukocytes, the white blood cells. Now, we have the thymus as well. And the thymus is going to be a unique organ because, as we saw with endocrine, it's going to be a relatively large organ, relative to the size of the individual in young children and newborns. But as we age and as we develop and grow, it becomes increasingly small and is ultimately really replaced just by fat or connective tissue. Okay, so sometimes in gross dissection in graduate school, when I did gross dissection, it's really, you really don't even see it, and you may just accidentally remove it with some adipose or fat tissue or some connective tissue, because it's not something that's super distinct in older individuals. It is in younger children and newborns. But what it is, it's a bilobed organ. It somewhat resembles the thyroid, but it's located a little more inferior in our body. And what it does is it serves for the maturation, the site for maturation of our T cells. So remember, the T in T lymphocyte or T cell stands for the thymus. This is where they're going to mature. So where did they originate? Where did they come from? Well, they came from the bone marrow, and then they drift to the thymus, and they mature there. Now, in the immune system lecture, we discuss how you can select self-attacking T cells to be destroyed so that we don't get autoimmune reactions. This occurs in the thymus. It's called, you have negative selection and positive selection. But for now, just understand that the T cells are going to originate in the bone marrow, and then they're going to actually mature in our thymus. Okay, so you can see the thymus. So this thymus relative to this heart back here, you can see this is it's relatively large compared to the size of the heart. So do you think this image on the left, would this be of an older individual or a younger individual? Hopefully you're saying a younger individual. And then on the right here, you can see the whole size of the heart within its pericardial sac, and you can see that the thymus is relatively small. So this would likely be a somewhat older individual, because you can see the size of the thymus relative to the size of the heart is different in these two. But it's generally located, you would look for it in the same place, kind of near the base of the heart on the anterior surface. So here you can see it again. In a newborn, quite large, covering most of the heart here. So you can see relative to the size of the heart, the thymus is very, very large. In an older individual, an adult, you can see that the thymus is very small. It has been replaced largely by fat and connective tissue, and is not near as large as it was in a younger individual. So that's going to conclude our lecture series on the lymphatic system. And so if you have any questions, please let me know, and I'll see you in the next video.